First, I would like to thank Sarah Teckman's wonderful introduction, and thanks for Sten Linnarsson and invitation and wonderful organizing committee of Human Cell Atlas. And it is really my great honor and pleasure to share my lab's work in the cortical development in single cell resolution during the past year. My title is The Diversity of Neurogenesis and Evolution in Human Cerebral Cortex. One of the most remarkable feature in the evolution of the neural cortex is enormous increases in neuron number that reach its peak in the human brain. Although the lambda organization of the cortex is relatively similar in all species, all mammal, an enormous expansion in cortical surface area underlies the transformation from a smooth cortex to the highly folded primate neural cortex. And the associated alteration of the cortical architecture that is the substrate for the high cortical function that distinguishes human from other mammalian species. This transition underscores the importance of understanding the process of neurogenesis in the developing neural cortex. How might the neural cortex become expanded in primates? So if we think about what could have happened in evolution to increase in the neuron in primate and human, we can think of this at a two level based on this scheme. First, we can we could make a lot of more neuron by first increases in the number of founder radial glia cell here, so we can get an expansion of the founder population. So thus, we can generate a large ventricular zone. Because the radial glia cell generally occupies the ventricular zone, this model predicts that you might have a large ventricular zone in developing. Second, if we can also make more neuron by increasing the number of round of transient amplification, so that is the intermediate protein cell can divide multiple times, accumulate in the subventricular zone before becoming neuron that migrate away. In this case, you could predict a large subventricular zone here, and all this you can see the movie. The intermediate protein cell, or we call the transient amplified cell, divide asymmetrically generate two daughter cells. Lately, there are another neural stem cell, or we call outer subventricular zone protein cell, either called ORG cell or basal protein cell, actually oriented from radial glia cell, translocated their nuclear to the subventricular zone, predominantly the OSVZ region from mitosis. So in many gyrocephalic mammal, the SVZ can be divided into inner or outer layer, which are separated by inner fiber layer, and different in terms of gene expression and the complement of neuron protein subtype. The OSVZ compartment became very prominent in primates, including human, and contain detached ORG cell. Recently, when some people also call the basal protein cell. And the movie actually show they have a translocated mitosis behavior. They jump and divide. So this kind of the behavior actually either expansion their proliferating zone, or either actually expansion their proliferating capacity. So thus, we could also probably postulate that the remarkable enlargement of the OSVZ and its addition of the neuron to the superficial layer is critical for the 1,000-fold expansion of the cortical surface during evolution, and might be the main cause of cerebral gyrification in carnivore, non-human primate, or human. That's to summarize here, dividing embryonic during the embryonic development. Excited neuron of the mammalian neural cortex oriented from a proliferating epithelium of radial glia cell that lie in the ventricle, cerebral ventricle. Much of our understanding of the even intervening with the radial glia cell proliferating and the neuronal production is based on rodent study, where radial glia cells are primarily restricted in the ventricular zone and undergo multiple rounds of asymmetric division to produce intermediate protein cell. While radial glia cell display an apical basal polarity typical of neuron epithelia, RP cell or intermediate protein cell appear to lack apical basal polarity. IP cells subsequently occupy the subventricular zone and divide asymmetrically to general neuron, and then migrate along the radioglial fiber to the pure surface, populate the cortical plate. While the thickness of the subventricular zone relatively constant over the course of neurogenesis, the OSVZ grew gradually thicker, reflecting a dramatic increase in number of ORG cell or basal protein cell. There are indeed a peak stage of neurogenesis. The OSVZ progenitor actually harbor up to 4 times as many as the ventricular and subventricular. So thus, right now, actually, we turn to be know about there are 3 major kind of protein cell for cortical development in the embryonic stage, like a radioglial cell, intermediate protein cell, and the OSVZ protein cell. So to investigate the diversity of the origination of the radioglial cell during early organogenesis, actually, luckily we analyzed a single cell transcriptome profile of whole embryo at post PCW 3 to 5. We have more than 83,000 cells clustered into 35 cell types, which were characterized into 11 lineage. This work was done by my collaborator, Professor Qian Wu in Beijing Normal University, and my assistant, Dr. Burton, and also my talent graduate student, Zeyuan Liu. So you can see clearly there are 11 different lineage cross, and also majority, you can see the PNS lineage and the CNS lineage down here. The nervous system developed earlier with 2 clearly distinct lineage here, the central nervous and the paranervous system. Since our data includes the same sample as early as the one at Carnegie stage 10, when neural tube formed, so we found a transit cell called neural mesodome progenitor here, which contribute to the development of the spinal cord, and adjacent paracel mesodome exists only at PCW as early as PCW3. Neural epithelial cells were predominantly observed from PCW3 and PCW4. We found that the neural epithelial cell divide into 3 clusters and regionalized along the brain cranial caudal axis, and as a forebrain, midbrain, and hindbrain, and the spinal cord progenitor showing spatial information. Interestingly, a few type of neuron could be found as early as PCW 3, 4, 5. You can see from here, especially for the sensory neuron and the spinal cord motor neuron, which show up very early at this stage. To further investigate the neurogenesis in early brain development, we also end the sample from PCW4 to PCW12 with our data from the last slide. Through totally 246,000 CNS cells in total, we perform velocity analysis and find that the neuron with different trajectory were from distinct radioglia subgroup, like radioglia 1 to radioglia 2, 3, 4. The differentially expressed gene in different radioglia subtype indicated the location of this cell, and including dorsal and ventral forebrain, midbrain, and the spinal cord radioglia cell. So definitely, so from this data, we pull out the regional defined radioglia cell. The number of the diversity of neurons dramatically increased in primates, especially in human, which indicate that might be some difference of human neural progenitor compared to rodents. So we next investigate the developmental similarity and differences between human and mice. By CCE, we found that cluster 13 here, and cluster 10 were human dominant, which were subset of the human neural epithelial cell and radioglia cell, respectively. Monocle 3 analysis suggests a trajectory relationship between cluster 13 to cluster neural epithelial to cluster 10 radioglia cell. So that's actually we figured out there are even the radioglia cell, they still have a spatial regional defined radioglia cell subtype. How about for the intermediate present cell? For intermediate present cell, why we call the transient amplified cells, because they specifically express TBR2 gene, T-box transfer factor, and the RP cell are derived from radioglia cell, and they divide symmetrically to double the number of the cell. So that's the reason we call the transient amplified cell. So in mice, the intermediate present cell relatively pretty small, in a small ratio. Morphologically, RP cell also have a very short process, resembling the philopodia and the neurite that translate contact other cell like a radioglia cell to mediate delta notch signaling. In mice, intermediate present cell usually generate a clone of 1 to 12 post-mitotic neuron. Well, with the evolutionary different species like Fishell, in Chinese Fishell, its evolutionary is very closely to the primate, although they look like a squirrel. Their brain were major lissencephaly. Interestingly, we found the Fishell have a similar amount of radioglia ORD cell with mice, but they will have a much more intermediate present cell, which have a high capacity of self-renew and neurogen, which suggests during the evolution, if we want to increase the number of neurons, we also can depend on the subventricular zone intermediate present cell to increase large number of neurons for cortical development. Then how about the RPC in the human, intermediate present cell in human? It is known that in human, the cortical RPS could divide many neurons to expand the progenitor pool, which were absent in mice. When we investigated the single-cell transcription profile for human developing cortex, which were mainly done by my talented graduate student Sui Juan and Xiaoying. Now Xiaoying is an independent PI in the Guangzhou Bioland laboratory. We found that the developing trajectory of NPC following three major paths. The early path is from radioglia cell to RPC cell. Second is from radioglia cell to ORD cell, and the third is from radioglia cell to ORD cell, and then back to the intermediate present cell. This indicates the origin of the EPC are either from radioglia cell or from ORD cell in human. So next, we assess the relative expression of UMES in pseudo time. We found two waves of intermediate present cell generation. To validate this single cell iron-seq result, we immunely staining cortex of the prefrontal from GW8 to GW19. In human, we observed early increases in the number of IPC at GW8 and just above the thick ventricular zone. And at GW10, the number of IPC increased rapidly in the expanded SVZ. Therefore, thereafter, RPC were not generated actively until GW16 when they were abundantly in the ISVZ and OSVZ. Our data suggests that there are two waves of IPC or intermediate present cell generation from radioglia cell at the earliest time and ORD cell at the late time. Both single cell iron-seq and in situ validation show that diversity of the cortical RPC in human. We also discover several new markers for the intermediate present cell. In collaboration with Professor Jie Qiao and Fuzhou Tang in Peking University, we also investigated the regional differences of human developing cortex. Surprisingly, we found a new subtype of the neural present cell we call NPC3, which are highly expressed in RPC marker and also neuronal marker. We believe this cell represent as a group of progenitor might at a transit stage, which show neuronal differentiation potential during the human brain development. So when signaling molecule were highly expressed in this transit state progenitor, and which is might suggest there are some specific signal pathway might regulate this specific progenitor cell. ORD, as we talked about from the introduction, ORD is considered as a basal predator, which contribute to cortical expansion and folding in primates. So to find the ORD specific gene, and is always a key question to us. So we combine microdissection, flow cytometry, and a single cell quantitative PCR and sequencing. We have reviewed a molecular property of the ORD cell. This work was done by my talented graduates Jing Liu and also collaborated with Professor Fu Chou Tang in Peking University. From here, we found very interesting one of the TM gene we call TMEM14B, which is validated by in situ hybridization localized measure in the OSVZ region. But also we found a couple of other markers you can see from here. The expression pattern also show different diversity pattern of expression, which might suggest even the ORD cell might have different subtypes. And this gene, one of the genes we call TMEM14B, which is evolved in the pretty high, in the especially in the oldworm monkey. So we conditionally express TMEM14B in mice neural predator. We found that cortex not only expanded but also show a bumper and a grooves. With detailed analysis, we found TMEM14B expressed in mouse brain generated at OSVZ like germinal zone and folded cortex with normal neural lamination. So here is an example showing that we can use single cell iron-6 to reveal ORD gene and play an essential role in control cortical expansion and folding. ORDs in cortex have been known for decades, like contributed by a great scientist from UCSF, my previous mentor, Arno Christian, and also William Huttner in Germany. So how about other brain regions? These were done by Xin in my lab. We searched and found in the hypothalamus, there were a group of cells show similar morphology to cortical ORD, and we named them hypothalamic mental zone radioglia cell. When these cells divide, they also undergo mitotic somal translocation. The majority of the daughter cells were neuron, and they were from hypothalamic radioglia cell. We found this human radioglia cell and human mental zone radioglia cell in mice and human. And this cell, you can see, they actually divide behavior similar mitotic somal translocation behavior. Then we actually further subset the HPC hypothalamic predator cell. We found HPC1 here, and look at this molecular similarity of this cell to human cortical radioglia cell and ORD cell. We found HPC1 to 3, which is correlated highly related ORD cell, but they still have some molecular differences here, which suggests the even in the hypothalamic region, they might also resident one of the similar behavior, mitotic behavior, ORD-like cell. Other than human tissue, we also developed a 3D culture system of human cortical organoid with vascular system derived from a HUVEC cell. In brief, by single cell RNC, we found that we organoid could mimic human cortical development very well in terms of all cell types and the transcriptome profile. This was done by my wonderful assistant in Chao Shi and my assistant Mengdi Wang in my lab. Although there were not very much cell type differences between organoid and V organoid, the ventricular organoid grew faster with more progenitor cell. You can see from the pseudo time analyzed, and the neuron matures earlier than the one in non-vascular organoid. Vascular help to reduce the pulp there here, you can see, and also cell death from our analysis. So I would like to summarize here. We found regional specified radioglia cell in the early nervous system development, and we found actually evolutionary conservation and the differences between mice and a human organon. So we also found a regional specified ORG, including their morphology, lineage, and distinct molecular feature. And we found the subtype and evolution of IPC, especially the diversity of IPC, they might play an important role during the evolution in some special species. And we also found a group of previous identified transit state neuron progenitor, which harbor both intermediate cell property and a neuronal property in the same cell. So I would like to stop here by acknowledge my wonderful collaborator, Professor Fuchu Tang at Peking University, and my Professor Jie Qiao at Peking University, and Professor Bing Liu at Fishell Medical Center, and Professor Shaorong Gao at Tongji University, and Professor Jun Zhang at Capital Medical University, and my longtime collaborator, Dr. Qian Wu at Beijing Normal University, and my good friend Tian Xue at University of Science and Technology College, and my friends Zilong Qiu at Institute of Neuroscience and Chinese Academy of Science, and Yunong Li at Peking University, and Yungjun Wang at Capital Medical University. I have to mention, I also I belong to State Laboratory of Brain and Cognitive Science, and I also belong to CAS Center for Excellence in Brain Science and Intelligence Technology, which were led by the world famous professor Dr. Mu Ming Pu, and also I would like to thank my brand. But I also want to mention, also we deposit all of the data in here, www.wanglaboratory.org. For all of you who are interested in our data and the story, you are very welcome to visit my lab website. We have built up an interactive data view at this site. This is lab fun during the pandemic stage. I would like to stop here. Thank you very much.
Fantastic. Thank you, Xiaokan. That's awesome talk, and the website is really fantastic for sharing as well. So yeah, thank you. Okay, so we move straight on now to Kimberly Saletti. Kimberly has been a postdoc in the Linnarsson Institute for the last 3 years, and we have her talk up next, and then we move straight seamlessly into the third talk by Nirmala Ryan, and then we have the panel discussion. So without further ado, I will hand over to Kimberly from the Karolinska Institute.
I'm just going to give a very brief discussion of the recent work from the group about the developing mouse brain. So a couple of years ago, the group published a cell type atlas of the mouse adolescent nervous system. So they sampled regions from throughout the central, peripheral, and enteric nervous systems in 3-week-old mice and performed single-cell RNA sequencing. And after analyzing the data to identify the cell types, as you can see in the dendrogram below, they found quite a diversity, especially in the neurons. And interestingly, neurons seemed most similar to other neurons from the same developmental regions, such as hindbrain neurons looking more similar to hindbrain neurons. And you can see this in the structure of the dendrogram. So interested in how this diversity arises, we undertook a similar effort in the developing nervous system as well. And so the sampling strategy that we used is shown here. So we sampled between embryonic day 7 and embryonic day 18. So on the graph on the y-axis, you can see the time points, and on the x-axis are the number of samples that we took for each of these individual time points. And for different ages, we used a different dissection strategy. So at E7 and E8, shown in the pictures below, we we dissected out the head and embryo. At E9 to 11, we dissected the brain into hindbrain, midbrain, forebrain. And as the forebrain expanded, we then further dissected the forebrain into multiple regions. And so we this resulted in about 300,000 cells with about 2,000 genes per cell. So this data is very difficult to visualize in just a two-dimensional way, but we took some inspiration from sort of old maps of the world. And so shown here in the center is the T-SNE of all the cells, colored by the major cell types like radial glia and neurons. And overlaid are contour lines that suggest roughly what's the cells inside the contours age, what embryonic age these cells came from. So you can see that on the bottom we captured some of these early embryonic cell types such as mesoderm and endoderm. And then as we moved into E8 and E10, we also capture neural crest and the radial glia. And then eventually we capture the development of neurons, and closer to E16 and E18, we start to see that the radial glia transform into a more glioblast state, and that seem more likely to give rise to astrocytes based on their gene expression. And wrapped around this T-SNE, we can see the dendrogram of the 942 clusters that we found. Most of the dendrogram is dominated by neurons, suggesting that you see a lot of diversity in the neurons, in comparison with just a small, I'm sorry, I'm unable to see my mouse, so I can't quite point this out for you, but the radial glia take up a much smaller proportion for on the dendrogram. And we're able to fit a bit more information to the corners here, so we can see so we can also plot cell type markers of interest, age to give a sense of the developmental progression, region, and so we can see that the forebrain cells, particularly the forebrain excitatory lineage, look quite distinct from the other neuronal lineages. And we can also plot a cell cycle score, which makes it quite clear where we can actually see the cycling progenitors in neuronal development. And so we've pulled out a lot of different subsets of cells and explored the data. I'll highlight here a group of cells that we consider to be organizers based on the expression of ligands that we know that to play a role in shaping brain development. And we were interested to see actually quite a transcriptomic diversity in these cells that hadn't been described before. So for example, we can see that floor plates in the midbrain and hindbrain are molecularly distinct, and these different organizer clusters also express different ligands, which has implications for sort of the regionalization of brain development. So I just want to also point out that you can explore the data yourself in this portal at mousebrain.org by clicking around on this visualization that I showed. And I'll end by acknowledging the rest of the group and collaborators and funders. Thank you.
Thank you, Kimberly. You've got us back on track on time. Much appreciated. And so next up, we will have Nirmala Ryan. Nirmala is in the Cheyenne lab, I believe, if I've understood correctly. She's the program leader of scissors in the Laboratory of Systems Biology and Data Analytics at the Genome Institute of Singapore. Over to you, Nirmala.
Great. Thanks for the introduction. Without wasting any time, I just want to start by thanking the organizers for having me on this forum. Happy to be here. So today I want to spend the next 10 minutes discussing about one of our studies that deals with understanding antidepressant action in the different regions of the brain. And the single cell section is a preliminary and ongoing work that leverages and builds up on a completed work wherein we surveyed 27 brain regions using bulk assays. primarily RNA-seq to understand transcriptome changes, and ChIP-seq to study epigenome changes. So before I dive into the data, I wanted to give an introduction into the motivation. Sorry, I seem to be having some problem. Yes, there we are. So we all know, needless to elaborate to this community, depression is a major public health concern, and the etiological factors are multifactorial, and depression affects the brain both structurally and as well as metabolic changes. It causes many metabolic changes in the brain. And though there are many molecular correlates associated with the neurobiology of depression, dysregulation of the monoaminergic system is the most widely implicated one, wherein the healthy brain has higher levels of serotonin in the synapse. And in the depressed brain, the serotonin levels goes down, and the action of the antidepressant is to bring back the level of serotonin and serotonin signaling. So the standard of care in depression is the first line of defense is SSRIs, known as the selective serotonin reuptake inhibitors, which work by affecting this SERT transporter. So it blocks the SERT transporter and thereby enables lots of serotonin levels to be available in the synapse. And of the many serotonin SSRIs that are available, serotonin reuptake inhibitors, fluoxetine or Prozac is the most widely prescribed one, but it has its own adverse side effects. And so therefore, there is a big need to understand the SSRI action and to better it. So there are some existing limitations in the current literature that we have. So we have a handful of studies that look into the genome-wide changes, but majority of them are based on one gene, one pathway, and really limited to a few regions. So we wanted to address this by doing an unbiased and a systematic analysis, but combining two rodent as well as primate models. And we specifically asked 3 questions. So which is of a broad panel of brain regions, which is the brain region that gets most affected by fluoxetine, which is the cell type that's most affected, and what's the molecular pathway. So with this in mind, we address these questions using multiple assays. We do bulk RNA-seq on 27 regions, ChIP-seq, and then for single-cell RNA-seq, right now we've done 2, and we expect and hope to expand the number to many more brain regions. So at the end of it, we'll be able to answer the 3 questions that we set out to answer with the region-specific fluoxetine changes that we observe. And so therefore today, I'm just going to talk to you about the single-cell RNA-seq study, a brief introduction into the results. For one of time, I will skip this. So just to assure you that we have high-quality cells, we implemented QC, and then we ended up with high-quality cells. And what you see here is that I used a reference-based panel, so a supervised clustering, and mapped on my single cells onto a reference panel, a mouse brain reference panel here. And we identified the major clusters. And then from there on, these major clusters, we sub-clustered them. We sub-cluster the major clusters into individual cell types. So RCA is a supervised clustering algorithm that comes from the lab, and Dubstepar is another feature gene selection algorithm. Both are available on GitHub for use. And so we ended up with single-cell RNA-seq data from 2 brain regions, the dorsal and the ventral dentate gyrus, which are the hippocampal zones of neurogenesis. And then we have about 16 to 17 subtypes. And so the first question that we asked, the very basic preliminary question that we asked is, do we observe reproducible cell proportion changes in response to fluoxetine across the different replicates? And just if I can highlight your attention to just these 2 cell types, the excitatory neurons and the OPC subtype. We identified that between the control and the fluoxetine group in dorsal dentate gyrus, there's a reduction in the excitatory neurons, whereas in the ventral dentate gyrus, there's an increase. So what we see here is, why is this exciting for us? Because very recently in February 2020, there was a paper that came out that looked at the single cell transcriptomes of depressed disorder brains, and they identified 2 populations, the OPC as well as the excitatory neurons. So validations are ongoing to further build on this simple finding that we have found. And so, but this gave us a lot of promise that there is just not regional heterogeneity, there's cellular heterogeneity in response to fluoxetine as well. And so therefore, the next simple question that we asked was, which is the key responder cell type? So there are different ways to address this question, but the one that we the low-hanging fruit was to look at the number of differential genes. And so what this plot tells you is that there are a lot more ventral dentate gyrus cell types that are affected by fluoxetine than compared to the dorsal dentate gyrus. And besides, it's just not limited to neuronal cell types. A lot of oligodendrocytes and astrocyte, a glial cell line, glial cell types are affected by fluoxetine. And we surveyed our DE genes, we investigated our DE genes to look for GWAS hits. And if I can draw your attention to one cell type, which is a neuronal cell type here, that's the most affected in both the dorsal and the ventral, that's most enriched for the SSRI GWAS. And besides that, you also see Bergman glia cells, which are enriched for SSRI response GWAS hits. And we wanted to verify some insights from bulk analysis. So from our bulk RNA-seq, we identified that in the dorsal dentate gyrus, certain neurotransmitter receptors are downregulated. But we wanted to pinpoint this downregulation of a neurotransmitter receptor to a particular cell type. And then we used our single cell data to exactly do that. And we identified one neuronal subtype, mature neuronal subtype, that shows its downregulation. With that, we have ChIP-seq results as well. So I'm just going to show you one simple example of how we kind of integrated both ChIP-seq as well as we used our single-cell RNA-seq to look in deeper. So while we were looking at a histone acetylation peak for K27 acetylation across 27 brain regions, we identified a very specific cluster that's very specific only to dorsal dentate gyrus. And that particular cluster was enriched for neuro-D1 binding motifs or binding targets. So we then looked into our bulk data set, and then we found that the neuro-D1 levels as well as its target, which is BDNF here, is upregulated in the dorsal dentate gyrus in the fluoxetine treatment compared to the sham. The upper panel is a control, and below you see the fluoxetine treatment. So we then looked for which subtype. We wanted to ascribe it to a particular subtype. So likewise, we found that one neuronal progenitor subtype could be responsible for this upregulation that we see. It's specifically upregulated only in this neuronal subtype. And so right now what we are doing is we are validating these some of the findings as well as the pathways that we have found by integrating both the bulk data set as well as the single cell data set that we have. And so I hope at the end of this, I can convince you that we have a genome-wide landscape of fluoxetine changes, both bulk and single cell. We have about 300 differentially altered genes at a single cell resolution. We hope to validate the key cell types and the molecular pathways. And we have some works ongoing where we are trying to integrate human samples along with our primate and rodent models as well. With that, I would like to thank my mentors. So I had phenomenal and inspiring mentors, both Shyam and Michael Meany. Michael Meany is based in McGill and Shyam at Genome Institute of Singapore. I would like to thank also my lab members for the fruitful discussions that we've had, and everyone out there listening to this talk. Thank you. With that, I'll take any questions if you have.
Thank you, Nirmala, for a fantastic talk and a different perspective on brain and stimulation responses. I'd like to now open up the panel discussion. We've only got a couple of questions on Slido, so we can also ask each other questions, and maybe the audience, I encourage the audience to basically submit questions on Slido, and we may try to figure out whether there's a way of also adding the audience into the panel as a single Zoom directly. So at the moment, we have a question that was actually asked during Xiaokun's talk, but I think it really relates to all three of you. And the question is how the work impacts regenerative medicine. So in other words, I think the way I would look at this question, I don't know, whoever submitted is anonymous, but the way I would interpret this question is, what can we learn from the in vivo development data for the in vitro systems, and how similar are the in vitro systems to in vivo, and how can we quantify that similarity? So I'll pose it as a broader question. Xiaokun, would you like to start if we sort of go backwards in the order?
Yes. I think this is a great question, and that is always we are think about when we perform the single cell multiomics analyze work when we were performed, especially for collect tissue from a human directly. And the way it's we right now focus on the early developmental stage is we really want to understand what kind of neural stem cell are there different type, why how we later we can use this in vivo developmental mechanism to regulate or supervise the embryonic stem cell in vitrally differentiate into different subtypes of neuron. Because right now we can see clearly they might have some regional specified neural progenitor cell or neural stem cell. They might generate or self-fate predetermined or later on be manipulated by environment to work together to deliver the neural stem cell to the neuron. But when we use that mechanism to culture in vitro, we have to think about very carefully what kind of neuron or what kind of neural progenitor cell we want to transplant later for the embryonic stem cell way to use it for for cure or cell therapy cure or something like that. So that's the thing we really call our attention, try to identify the diversity or specificity related to regionalized or the difference of radial glial cell or intermediate cell or RG cell. That's I think that will give a lot of information, and previously we will never understand for especially for cell therapy, for embryonic stem cell therapy, for especially for brain development, for brain disease. That's what one of my major drive force for we peer really into the human early developmental stage. Try to understand the mechanism or regional diversity of the neural stem cell or neural precursor. Yeah. For evolutionary way, we also want to know what kind of evolutionary differences are they conservative mechanism or divert when we evolve into the human brain human. What kind of the major mechanism to drive this cell? How they divide, how they differentiate to build up our high cognition human brain. Well, I think one of the that's also very important to understand for the medicine study. That's what this is two major concepts. Yeah.
So basically, if I understand you correctly, you're saying that there's the kind of molecular details of which factors are required to control kind of the differentiation of the different lineages, cell signaling, and so on and so forth, and to mimic those in the dish. But then there's a bigger question about consciousness and the overarching function of the organ. So if we start with kind of the signaling factors and the molecular kind of level of differentiation of the different cell lineages, how far do you think we are towards creating faithful cells in the dish for the different neuronal lineages? I mean,
I think
what are the big areas that are missing, or how far away are we from being able to engineer?
I think especially for the neuronal differentiation or neuronal development, one of the major obstacle right now is can we recapitulate the functional or the firing pattern of different subtypes of neuron in dish, which we could use it for later for medicine or for cell therapy, and whether or not there are cell type specificity supervised neural circuit formation, which is also very interesting. Whether or not we can, but if we think about, we have a lot of innate behavior like a thirsty, like a food, like a hunter. It's innate behavior which is imprint. We might lot of behavior we know from the we give in birth. We give birth. We will know that. So a lot of the innate behavior, especially inside the brain, especially for the hypothalamus region, might have some gene DNA coded or synthesis encoded or cellular subtype coded, and whether or not can we find out like a specific cell type supervise specific neural circuit formation. which is actually right. I didn't have time to talk about in this today, but we do perform a little bit. We try to perform specific behavior, and we in vivo recording from the mouse brain, and we can pick up the cell with 2-photon supervised in vivo patch seq, and we can pick up the cell. We can sequence it. We do find there are some cell type specific, molecule specific all-trusted neural circuit activity. So I guess they might have some they might have some basic mechanism inside, but in terms of like high cognition, I think we still need there's still a little bit far way to go there. We need a more advanced technique. We can combine, for example, like high resolution functional MRR combined with the single cell multi-omic sequencing.
Yeah. No, I agree. Yeah. From a functional readout point of view.
Yes.
I see. I understand where you're coming from, and what function means in neuroscience is the circuitry, the electrophysiology, and then ultimately the consciousness and these larger. Nirmal and Kimberly, would you like to add to this discussion about, how the data informs regenerative medicine? Kimberly, are you? What's
sure,
your perspective?
I think I agree. It's very important for understanding the molecular regulation of everything, just as that was already so nicely said. I think maybe I could emphasize that I think these sort of atlasing efforts are really important for putting these molecular programs, I guess you could say, in context. So it's sometimes a lot of these, many of these transcription factors, for example, are reused to perform different jobs during development. And I think getting an overview like we have in our data of what's happening in both the forebrain and the midbrain and the hindbrain allows us to put these transcription factors in the backgrounds that will lead to various cell types and understand that aspect of development. So I think that these sort of comparative efforts of different stem cells and really getting an overview of the entire brain is useful for regenerative efforts.
Awesome. Nirmala, would you like to contribute to the discussion?
Sure. I completely agree with Shaukan and Kimberly said. Maybe I would just want to add on to one point, like besides cataloging and the molecular integrations that we do at a single cell level, I think eventually we would also move on to what Shaukan touched on, functionally validating the models, like doing single cell functional assays. I think we are getting there. From beyond RNA-seq, we reached ATAC-seq, and now we are doing single-cell proteomics, metabolomics, and very soon we'll be doing high-throughput, probably drug screening, or that would inform regenerative medicine as well.
Awesome. Thank you, all three of you. I'd like to move over to some questions for Nirmala, which are whether the fluoxetine response will be different in diseased brains, and whether you can mimic, for instance, like a depressed brain versus healthy reference. So that's the first question. And the second one is whether you could explain the rationale behind the choice of the small molecules that you were using.
Got it. So to address the first question, is this model system valid? So the model system that I'm using are naive rats. So historically, if you the pharmacological validation or the understanding SSRI functions have always been done on naive rats for one reason. The major reason that which model system would you pick, which depressed model would be better, right? There are many models of depression. So the question would arise, which is a better model compared to the other? So a safe start would be to work on naive rats, and from there on, move on as a next step to a depressed model, or to cross-validate with a depressed model. I did not show you data sets wherein they exactly did that. So I took the D genes and the DA peaks, the differential genes and differentially acetylated peaks that we identified from our bulk studies, and we cross-referenced it to a depressed model that was also treated with SSRIs that we had done as an unpublished data with fluoxetine as well as a couple of other SSRIs. And we do see a good correlation there. So that's promising and encouraging. So the second question was, sorry, remind me, Sarah, the second question again.
No, it was basically why the rationale behind the choice of the medication or small molecules. I mean, it's clear because they're used in the clinic, right? Is that correct?
That's right, yeah. So we chose fluoxetine because 250 million people are on antidepressants and 64% of them are on Prozac, which is fluoxetine, which is precisely why we chose it. In parallel, we've also completed a study where we compared different SSRIs with non-SSRIs as well. But that's not shown here. That's unpublished data. Yeah.
Fine. Then there was a question about whether animal models of depression can be chemically induced. But I mean, all your model is basically using human cells, right?
My model is we have rodent models where we treated rodents.
Okay.
Yeah, we have rodent models where we treated rodents with fluoxetine for 8 weeks, and then we have primate models where we treated them with fluoxetine for 8 weeks, and then we hope to go on into human postmortem samples, single nuclei analysis of human postmortem brains, where we compare depressed versus those who were on SSRI, depressed plus SSRIs. The question was,
the animals aren't depressed. I mean, the animals are treated, right?
The animals are treated. They are naive rats treated with fluoxetine. So,
okay, I think that was the question. So then there's a question for all three of you, and it's whether there's any plan to study the homeostasis of brain tissues at different developmental stages and adulthood, and whether stem cell migration can be captured within those studies. I guess more for Kimberly and Shalkan, probably.
Kimberly, you first. Okay. I think maybe I can go first. I think this question might relate to because neural migration, for radial cell generated neuron, they migrate radially, so it's easy to capture. For ORG cell, still we need a lot of effort to follow up whether or not the neuron generated from ORG, whether they migrate, whether or not they allow the radial glia the mother cells fiber. And for the intermediate prone cell, they just divide locally and they just migrate with the inside out way. But for the ventral part, especially for the GE part, ganglion eminence part, which majority generate interneuron, they migrate tangentially a long way to go. And it's if we perform the single cell iron-seq, it's difficult to capture the migrating neuron locally with the stem cell. That's I think maybe the audience try to ask. For that part of the work, I think if we still can take advantage of the powerful single cell iron-seq data, we can actually do the sort of like assay to project or predict the potential neural cell fate. Actually, I don't have time to talk about too much about this, but we have a ongoing project. We are trying to pull out the human interneuron stuff, and we actually can found the cell fate predetermined mechanism which supervise the new precursor, which we people call precursor, but we also call newborn neuron in the interneuron part. They actually can predict or project where they migrate to. So, well, still, definitely, we ethnically, if we can get a tissue from all kind of states, a donated state, that would be perfect. We can capture the migration trajectory or migration trail. But still, I think from the right now, we can pinpoint where they're going and how they can turn to be mature neuron, especially for the interneuron part. For excited neuron, I think it's much easier. We can just capture locally, and we can tangle the neuron. But for human, most of the neuron migrate might stop after birth, but still there are still neuron migrate, especially in the third gestation, which is I think very rare tissue. We can get a donation. So that's one of the typical part for that. Yeah.
Yeah, it's the same in the UK. Yeah. Kimberly, anything you'd like to add?
No, I don't think I have anything to add here. We're not planning to look at migration necessarily here. In our data set, we did sort of gross dissections of the major brain regions, which I think makes it difficult to think about sort of whether or not the cells are moving around. But certainly there's a lot of interesting experiments you can do, and it would be very interesting.
Yeah, I might want to end one. Maybe still we are trying to figure out how to do the organelle culture, fusion different part of brain together, like Professor Pascal did in Stanford. They can fusion culture cortex with the ganglion amylase part of the organelle, so they can figure out, they can see the internal migrate to the cortical plate organelle. So I think from that, we might can capture some mechanism for how neuron migrate. Yeah, that's one of the way to fill in the gap.
Okay, so we have. I think the migration question, is for me. I'm interested in the development of the immune system, and Maz Hanif and I have done a lot of work on the development across different organs of primary and secondary immune system. So I wonder whether, Maz, would you like to switch on your video and give a perspective from development of immunity, how we can look at different developmental stages and track the migration of precursors from the bone marrow to the thymus and so on. I think maybe that's a system where it's really very nice and easy to think about this, because the cells are happy to move, maybe more than neurons. They're happy to migrate around the body.
Oh, thanks, Sarah. Hi, everyone. I think it's I was very intrigued by this concept, because the brain is sort of almost seen as always slightly isolated, although in truth it's widely connected to the rest of the body, much in the same way as the immune system, because you've got nerve fibers running throughout the entire body. But the immune system is quite interesting from the perspective of it. You have different organs, and it's sort of like sequential acquisition of hematopoiesis function across these organs, beginning in the yolk sac, and then you've got the aortogonad mesonephros, the fetal liver, and then subsequently the fetal bone marrow. And then what we find during development is the kind of then migration of these cells, both progenitors and differentiated cells, into the peripheral tissues, where they then subsequently acquire the microenvironment of the peripheral tissue and function as an immune protection in those tissues. So it has similarities in the sense of where it's originally formed, and then the subsequent migration in much the same way as you have within the brain. And I was also struck by Kimberly's data. I think I posted a question there about some of the red blood cells that you see, the erythroid colonies, both primitive and definitive in the brain. And I was wondering how much of those are actually intravascular and how much of those are extravascular, and how these kind of like, in essence, distributed systems also interact during development. Would be interesting to hear kind of from the brain perspective, the kind of compare and contrast between the two systems, which are essentially distributed. Kimberly, maybe you could elaborate on that.
Kimberly, could you, because so you showed primitive and definitive erythroid clusters. And yeah. What Maz is asking, whether you know whether they're inside the vessels or they are sitting inside the brain tissue.
Right. Actually, I don't know. I'm not sure. I think based on our dissection, I don't have any good markers of landmarks based on to know exactly where they're coming from. And we also so I think a lot of those will come also from the early embryonic stages, where we pretty much dissected the whole head or the whole embryo. at embryonic day 7 and embryonic day 8. So I'm not sure actually. I don't know exactly where they come from.
Yeah, but there wouldn't be any in the head part. There wouldn't be any. I mean, the yolk sac and so on would be separate. So yeah. It would be interesting to know that.
But it would be also kind of like from the perspective of like, I mean, most of you are kind of studying the brain, the central nervous system itself, but do you think the peripheral nervous system, when it's innervating the tissues, much like the kind of immune cells, do they sort of adapt locally a bit more in the, you know, is that something that's recognized or thought about in the same way that perhaps during the CNS migrate within the actual, you know, cortical brain migration, you have adaptation in the different regions?
You mean whether the peripheral nervous system sort of develops differently, depending on where it migrates into, or like along the body, or?
Yeah, if I make it clearer, so if you have, like, for example, a lymphocyte, it goes into the skin, or it goes into the gut, or it goes into the kidney, it then adapts to that microenvironment and sort of has some transcriptional changes that are more suited to perform its function in those environments. When you've got like brain development, you've got the very different regions of the brain that are highly specialized. And that's somehow like a compressed version of that. But you've also got the peripheral nervous system where you've got nerve fibers in the skin, nerve fibers in the kidney, and nerve fibers in the gut. So is there adaptation in those settings, or, you know?
Yeah, I haven't explored developments in the peripheral and enteric nervous systems. So the adolescent data set captures some of these cell types at 3 weeks. I don't know too much about it earlier than that. But I think you can see specialization along the peripheral nervous system. But it would be of course interesting to know how that develops, and I know people are working on that, but we didn't capture those nervous systems in our developmental data set.
What do you think, Sharkan?
Yeah, I think it's a very interesting question, and well, we do have I mean, we can maybe later we can double check that, because we do have the whole number whole number of data from PCW 3 to 5. So there already have a little bit of organ genesis, but we didn't focus on that. So we just focus on central nervous system, and we collect tissue after 5. We just collect the head, and until 12 we collect just brain. So because it's too many cells, we cannot handle that. So we just use like a 12 weeks brain as an endpoint to try to pull out the trajectory, how the stem cell project to the neuron. But we do have we are not expert on this immune system on a very early stage, but we do have the data. Maybe we can talk about later. Like I can pull out some. We don't know the marker. If you can share some marker, we can pull out the cell for you. But I think it's very very interesting question, how the peripheral nervous system penetrate into the organ and how they coordinate. I think that's really a good angle to look at that. Yeah, but unfortunately we don't have expertise on that part. So yeah. But we can check later. If you like, we can just share the data. We can check later. Yeah.
Yeah.
I think that makes a lot of sense. Is a really exciting angle. Yeah.
Yeah.
Mats, did you have another question, or was there another question?
There are questions on Slido.
Okay, so there's one more that we haven't covered yet, which is for Nirmala. And what is the status of the BBB in patients with depression?
What is the status of the blood-brain barrier in depression, patients with depression? I'm afraid haven't worked on that to specifically answer the question. But based on the literature, I would say that there is a fair percentage of depressed patients that have compromised BBB, and that lead them to, so there's a neuroinflammation angle to the whole aggravation of depression, and that has been pinned down to the weaker BBB system. Yeah, so that's a few literature. There's a few publications in the last 5 years.
But what do you mean weaker? Do you mean like a breach or an activation of inflammation, or what do you mean by weak?
The paper that I'm referring to, I forget which journal and who the authors were, but the paper that I'm referring to looks at a small percentage of depressed people who have had a breach in the BBB with neuroinflammatory cytokines rushing in to aggravate the inflammation. Yeah, so that's what I was referring to. But personally, I do not have any data that talks about the BBB status.
Can I ask, following up on that?
Yeah,
actually,
please.
Do you feel that this is also relating to the time frame that you're looking at? Because, you know, in your model, the kind of like time frame that you analyze might be different to the sort of like fluoxetine effect, particularly in the long term in humans.
Yeah. So the time frame that I am looking at in our rodent models is 8 weeks treatment. Both in our rodent and primate models, we've chosen 8 weeks, precisely because that mirrors the time taken in humans for fluoxetine to cause a change. To see the clinical benefits of fluoxetine, it takes a long time in the humans. So that's exactly why we've chosen 6 weeks. We feel that 6 weeks correctly models it in the rodent system. And to address your question in connection with a BBB, was that your question? Like, does this 8-week treatment?
Well, I mean, it's more of like, presumably you don't see any inflammation, you don't see any influx of, you know, immune mediators or whatever. And, you know, often people take fluoxetine for much longer than 8 weeks. The 8 weeks is kind of like where you first, you know, where you tell your patients that, you know, you don't expect any response so early. Exactly. But, you know, but people kind of continue with the treatment for much longer than that.
That's true. That's true. That's true. Exactly. So I think we hope that the postmortem patient samples that we hope to receive from our collaborator would help us address that question, because we have categorized patients at different treatment time points. So the longer we would have samples which were on longer time versus shorter treatment times. So perhaps that comparison could help us understand that.
Yeah. There's a question on Slido relating to this specifically in terms of how would you model the external environment effects in comparing fluoxetine response in mouse and human, I guess.
Well, in the study system that we have chosen, there is no way that we could model the environmental influence on the effect of fluoxetine. But Michael Meaney's lab has precisely done that. Eric Nessler and Michael Meaney's lab have, in collaboration, done that. So they have looked at how cocaine and few other SSRIs besides fluoxetine are influenced by the environmental factors. Like, for example, environmental enrichment. If the mice are put in a novel environment versus with enrichment tools versus where they get to play versus a stale environment, how does fluoxetine effect change? But I have not done the comparison for fluoxetine in this particular study. That's something, and that's an interesting angle perhaps to look into for future studies.
Thanks. There's one question left, which we haven't discussed, and it's kind of for everyone. I don't know who wants to sort of like take the question on, so I'll leave it open. It's about the kind of some of the discussions about brain organoids and consciousness, and, you know, it would be great to hear the panel's view on this. Any offers? Maybe Shakun, would you be happy to give a stab at this question?
Well, I think especially for the consciousness, I think it's still too far to talk about that, because it's first is ethnically regulation, and we might don't want to go there too far. And the second is like currently the organoid culture is still just recapitulate very early brain development state, and we can see some like a firing pattern. We can see some like a calcium wave, but it's still just mimic the early brain development. For example, like from the very early beginning, the neural circuit, before they go into the very fine organization, they have like a calcium wave bursting in and kind of like a burst and a calcium wave, which is majority is electrical coupling between the neuron to neuron, which is not very well defined neural circuit. So I think still we have a long way to go to get the organelle might have conscious capability, but still I think still we can actually think about for the cell therapy, we perform like a transplantation for the human brain organelle to the mouse host, but actually when we transplant the organelle, we can see much less cell die compared to you transplant pellet, which might actually suggest it's one of the good way to transplant the chunk of brain-like tissue or a part of brain-like tissue, sort of like we call organoid, for the regeneration medicine. That's what but still I think it's far away from to really get consciousness brain organoid. Yeah, that's what I comment. Yeah.
Kimberly, have you any thoughts about it?
Well, I was thinking something similar, I guess, actually. I think I was thinking I haven't worked with organoids, but from what I understand, the cell type composition suggests that they're still quite early. So I was thinking as well that it doesn't seem like they're headed there yet toward consciousness, but it's really something to think about.
No, that's brilliant. Yeah, I mean, I think, those were the kind of like primarily the questions that have been posed on Slido. And maybe one of the things that, we haven't discussed is kind of trying to think about what do you think might be the kind of like key challenges that we have now in kind of having generated or amassed all of these data sets. Where would be, where should we be heading, and what are the kind of like current challenges that stops us from making this the progress that we want to make into either transplantation, curing disease, and so on and so forth. It's kind of quite a broad question, but it's useful to know in terms of what the community can do and what the community can support with to overcome challenges that the panel might be experiencing. Shakun, I think you kind of unmuted us. So.
Yeah, I think I really appreciate we have a human cell atlas organized a committee which is trying to group all the scientists from all around the world to work together to pinpoint the cell atlas from the early development to the adult stage. I think if we don't really understand what kind of cell is more valuable or what kind of cell we have being sensitive, I think it's still a little bit far away to practice medicine, right? Because it's especially for my little bit of experience about the brain tumor, we can see the diversity and the heterogeneity from every case by case, and even the tube tumor from the single case from single patient, they also diverge or heterogeneous very differently. So that really makes the cell and to fully understand the cell atlas, this is very necessary to really understand what kind of cell we have and what kind of cell go wrong and what kind of precisely molecular regulation mechanism, and also refer to specific small compound to deal with. I think that's still I think it's human cell, to understand the fully picture of the human cell map still in the early stage. That's really, I think, thanks Sarah Techman and we all kind of like scientists effort, really group people join together to share, to collaborate, to open access everything that we know. And I think that's a very good start. and I think it's very promising. I believe they're going to be, eventually we figured out, get some overcome the obstacle right now in very soon. That's what I really enjoy the human cell atlas stuff, and also I enjoy every meeting, and I want to, I want to contribute to that, and that's just what I have to talk about. Yeah,
that's brilliant.
Thank you, Jalkan. That's a fantastic way of wrapping up this panel discussion, I think. And I'm incredibly grateful to you and all the labs who are contributing data and insights and discoveries, towards building a complete human cell atlas. So I would like to thank the panelists, Jalkan, Kimberly, Nirmala, and Maz also for joining me in moderating the discussion and contributing to a fantastic discussion today. And thank you to the program committee, Bruce, Gary, Alain, Dana, Maz, Sten, Aviv, Diane, and also the executive office staff and Brian from the AV who are working in the background. So Christine Schwenk and Tracy Andrew from the HCA executive office have been masterminding all the organization. And thank you for your wonderful talks again. And thank you for everybody for listening and for contributing questions online on Slido. We had a great time. Thank you and have a good day, everybody. The next seminar will be on clinical and rare disease. And we have an exciting set of speakers and panelists coming up relating HCA Developmental Atlas to rare disease, pediatric disease in December. And the Asian Australia seminar will follow the next week. See you then. Bye.